Hello friends, I'm Archana Sachdev, Principal Scientist, Division of Biochemistry at Indian Agricultural Research Institute, Delhi. In today's lecture, we are going to talk about extremozymes and their applications, especially the adaptations and applications of halophilic enzymes, adaptations and applications of acidophilic and alkaliphilic enzymes, adaptations and applications of organic solvent stable enzymes, and applications of enzymes from piezophiles, metallophiles, and radiophiles. We shall also see the challenges associated with bulk scale production of extremophilic enzymes. This slide shows the concept map that will be followed in this lecture. We will first study about the adaptations and applications of other extremophilic enzymes like those from acidophiles, alkaliphiles, halophiles, piezophiles, radiophiles, metallophiles, and solvent-stable microorganisms. Later during the module, we'll also see what are the challenges associated with the bulk-scale production of extremozymes. Extremophiles are microorganisms living in extreme environments and naturally attuned to withstand extreme survival conditions like extreme heat, extreme cold, extreme salinity, etc. They often belong to the domain of archaea and bacteria. Extremophiles are perceived to be one excellent source of stable enzymes. Their enzymes are naturally stable and exhibit optimal activities even under harsh conditions. The adaptations in extremophilic proteins do not seem to follow any generic trend, but are specific to extremophile and the surrounding environment conditions in which it is thriving. Microorganisms that are able to thrive optimally in saline environments or saturated brines are classified as halophiles, and the enzymes derived from them are referred to as halophilic enzymes. Halophiles, which belong to the domain of archaea and bacteria, are usually isolated from salt-saturated brines, saline soils, salt lakes, seawater, salt deposits, and also from fluid inclusions within salt crystals, as shown in figure number 1. Halophiles are categorized as non-halophiles, which grow under less than 0.2 molar salt concentrations, slight halophiles between 0.2 to 0.5 molar salt, moderate halophiles 0.5 to 2.5 molar salt, borderline extreme halophiles 1.5 to 4 molar salt, and extreme halophiles 2.5 to 5.2 molar salt conditions. Halozymes, the proteins from such halophilic origin, are endowed with unique structural features and catalytic powers to sustain the metabolic and physiological processes under high salt conditions. Apart from their salt stability, they also exhibit ability to withstand other extreme conditions at times. Some of the specific structural features which these enzymes have evolved for greater enzymatic flexibility and catalytic function are enlisted as such: high acidic amino acid content on protein surface, low lysine content, relatively low hydrophobicity at the core of the protein, increased salt bridge formation, and these modifications impart structural flexibility to the halophilic enzymes and enable them to catalyze reactions efficiently under salt conditions. Halophilic microorganisms have been perceived as an ideal source of salt-stable enzymes. Enzymes derived from halophiles are mostly hydrolases, namely amylases, proteases, nucleases, cellulases, chitinases, xylonases, esterases, and lipases. In comparison to extensive application of psychrophilic and thermophilic enzymes, very few halophilic enzymes have found commercial applications in industry and biotechnology. Nonetheless, broad areas of applications of halophilic enzymes which are being explored are shown clearly in table number 1. Although not many large-scale commercial and industrial applications of halophilic enzymes have been seen, halophilic enzymes, as shown in table 1 above, indicate immense areas where they could be potentially used. and we can see from the table various halophilic enzymes have been investigated for potential applications, but their commercialization is yet to be achieved. While halophilic proteases are important for preparation of pickles, fish sauce, non-aqueous protein synthesis, water waste remediation, halophilic silonases and cellulases, they have also been found to be useful for paper industry and biofuel production. Other enzymes like amylases, lipases, galactosidases, lactamases, nucleases have also been explored for various applications like starch liquefaction, organic synthesis, lactose hydrolysis, degradation of lactams in molecular biology, respectively. Halophilic enzymes can also be used in biotransformation reactions at high salt concentrations, bioremediation of toxic compounds, decontamination, bioremediation of saline waste for pollution control. Alkaliphiles, as the name suggests, are microorganisms which require an alkaline pH of 8 or more for their growth. They usually grow optimally at pH 10. Most of the archaeal alkaliphiles are halophiles additionally. Alkaliphiles can be isolated from alkaline hot springs, alkaline hydrothermal vents, alkaline lakes and ponds. Acidophiles, on the other hand, are microorganisms which can thrive under low pH conditions, that is pH 3 or lower than that. Most of the known acidophiles are also thermophilic in nature and can withstand high temperatures. Acidophilic and alkaliphilic microbes have a unique property of being able to maintain a neutral pH in their internal cellular environment through the use of proton pumps. Thus, the intracellular enzymes in acidophiles and alkaliphiles are not necessarily required to have any ubiquitous structural adaptations, unlike psychrophiles, thermophiles, or halophiles, for functioning effectively and optimally. However, in case of extracellular enzymes, the case is different, since the enzymes are released into the environment, which has high or low pH, and the enzymes would need to function optimally under such environment. Although the exact molecular adaptations of acidophilic and alkaliphilic enzymes have not been explored in details yet, some of the present understandings are summarized below. In case of acidophilic proteins, the adaptation to low pH is attributed to the presence of an increased number of negatively charged amino acids on the protein surface. This subsequently resists destabilization of the protein or the enzyme induced by low pH. Other factors may also include reduced solvent access for these acidic residues and binding of metal cofactors to impart further acid stability. Protein adaptations to high pH in alkaliphiles may be attributed to some of the following factors. Number 1, increased number of negatively charged amino acid residues on the protein surface. Number 2, increased hydrogen bonding and hydrophobic interactions. Number 3, fewer hydrophobic residues exposed to the solvent. Although commercially unavailable, alkaliphilic and acidophilic microbes have been reported to be producers of several hydrolytic enzymes like amylases, cellulases, proteases, catalases, xylonases, pectinases, etc. These can be explored for potential applications, as has been discussed earlier. Some interesting areas of application for acidophilic enzymes are outlined below. For biofuel production, acidophilic cellulases and xylonases could be used for hydrolysis of lignocellulosic material and improved ethanol production. For bioleaching utilizes acidophilic microorganisms and their acid-stable enzymes for harvesting metals like copper, nickel, cobalt, zinc, and uranium from acid and metal contaminants, which are usually released from mining sites. In the food industry, where improved efficiency of monosaccharide production by acid-stable glucoamylases is performed. In the next slide, we'll take a look at the enzymes which are stable in organic solvents. These enzymes have laid the foundation of non-aqueous biocatalysis and are considered as extremely useful for industrial purposes which use low water, non-aqueous based reactions. Microorganisms which can grow in the presence of organic solvents cater to the category of solvent-stable extremophiles. Consequently, enzymes derived from such microorganisms will be attuned to function under low-water, non-aqueous conditions. Use of such enzymes for catalyzing reactions being performed in non-aqueous medium has paved the way for extensive application of enzymes in organic synthesis. Natural enzymes with organic solvent stability and tolerance are useful for applications as catalysts in non-aqueous reaction media because they can be used without any modifications to stabilize these enzymes. Adaptations of such naturally occurring solvent-stable proteins include the role of disulfide bonds, salt bridges in the proteins, and the presence of hydrophobic patches on the protein surface. The stability of these solvent-stable enzymes may also be attributed to the stability of their secondary structure. Use of enzymes in organic solvents has been exploited in various areas like non-aqueous peptide and ester synthesis, transesterification, galactosylation, kinetic and optical resolution, and synthesis of oligosaccharide propionates. Table 2 clearly outlines some of the solvent-stable enzymes, their sources, and their application areas. We have seen a plethora of solvent-stable enzymes which are produced from microbial sources. Their applications in biocatalysis of non-aqueous synthesis of organic compounds, non-aqueous peptide ester oligosaccharide synthesis, optical resolution, as well as biochemical reactions have been explored. Some such examples are provided in table number 2 shown here. Lipases and esterases, which are solvent-stable, have been primarily used for ester and peptide synthesis or transesterification, while amylases have been put to use for malto-oligosaccharide synthesis and proteases for peptide synthesis. The important thing to note here is that the organic, low-water, non-aqueous medium, these hydrolytic enzymes are indeed catalyzing the reversible reactions that lead to synthesis of ester, oligosaccharides, or peptides. Other than solvent-stable lipases, amylases, or proteases, several other enzymes like cholesterol oxidase, alcohol dehydrogenase, glucosidase, galactosidase have also been reported, which have been found useful for optical resolution, bioconversion, synthesis of enantiopure or regioselective compounds, etc. These are microorganisms which are able to survive under high pressure conditions, for example in deep ocean depths where pressures higher than 10 MPa exist. They are also known as barophiles. Piezophilic enzymes like hydrogenases and glycosidases have been isolated from microbial sources like Pyrococcus species, Thermus species, Shevanella species, Methanococcus, Photobacterium species, etc. These enzymes are also polyextremophilic in nature in that they also exhibit thermophilic and psychrophilic properties. The understanding of exact adaptive features of piezophilic enzymes are in the stage of infancy. The study of how piezophilic enzymes stabilize under high pressure conditions will enable us to design synthetic enzymes to increase catalytic efficiency of processes such as food processing and antibiotic production performed at high hydrostatic pressures. Although piezophilic enzymes, because of their ability to be catalytically active under high pressure conditions, reflect tremendous potential for use in biotechnology industry, practical industrial applications, however, are few. This can be attributed to the difficulties associated with culturing of these microbes in lab and industrial scale. As the name suggests, metallophiles are those microorganisms which can grow under very high metal concentrations in the environment. They can be isolated from metal wastes, sediments or soils from industries with high content of heavy metal. One example of metallophile is Ralstonia metallidurans, which has been shown to have genes for multiple resistance to heavy metals like zinc, cadmium, cobalt, lead, copper, mercury, nickel, and chromium. Enzymes from metallophiles have been found to be useful for heavy metal bioremediation purposes, although these have not been industrially explored as of now. Microorganisms which survive under extreme conditions of high ionizing and UV radiation are called radiophiles. Radiophiles can be isolated from nuclear plants or areas which are rich in nuclear waste. And examples are Deinococcus radiodurans, Deinococcus radiophilus, Thermococcus marinus, Thermococcus radiotolerans, etc. The mechanism by which radiophiles are able to withstand high radiation involve a unique arrangement in their cellular DNA, which enables a rapid repair of the DNA caused by radiation. This feature also makes them resistant to many mutagenic chemicals. Currently, there are no specific applications of enzymes from radiophiles. Given the difficulty to culture and grow radiophilic microorganisms on industrial scale, genetic engineering methods are being employed to express the desired genes from radiophiles in suitable hosts for exploring their applicability of these enzymes or proteins in bioremediation of nuclear wastes. Extremophiles have been shown to have significant economic potential in several industrial avenues which have been discussed. For example, food, detergents, textile, paper, molecular biology, leather industry, environmental, bioremediation, etc. The increased use of extremophilic enzymes in related fields in the years to come is thus evident. However, there are certain challenges associated with the production and purification of extremophilic enzymes. Now let us take a look at it. Some of the drawbacks associated with extremophilic enzyme production include difficulty to culture these microbes at lab or industrial scale. Extreme environmental conditions are required for growth of extremophiles, and they are often not available in the industrial fermenting conditions. Then lower growth rate of the microbes. Difficulties associated with expressing extremophilic gene in mesophilic host, for example, requirement of cofactors that may not be utilized by the host. Then concerns associated with purification of extremozymes. Such process would require extremophilic environmental conditions to be maintained during the purification process. Then challenges faced with expressing extremophilic enzymes in native hosts. The increased use of extremophilic enzymes in related fields in the years to come is evident. With the improved and advanced culturing techniques, purification strategies and development of molecular biology, and protein and genetic engineering tools, it will not be long before extremophilic proteins are better understood and tailored as per the industrial need. Thus, their availability and applicability at the industrial level will substantially increase. It is to be realized that a lot more in-depth search is essential to be able to have large-scale production of extremozymes and further obtain industrially relevant products through the use of these extremozymes in biocatalytic processes. It is envisaged that regular use of extremozymes in the industry will contribute towards lowering the cost of production, having optimized environment-friendly processes, and many other advantages. To summarize the lecture, we can say that halophilic enzymes are able to function optimally under moderate and high salt concentrations without getting denatured. Halophilic proteases, amylases, lipases, xylenases, cellulases, nucleases, etc. find potential applications which need to be explored on an industrial scale. Alkaliphilic and acidophilic enzymes are attuned to function under high and low pH respectively. Not many applications of alkaliphilic and acidophilic enzymes are commercially known. Organic solvent stable microbes are sources of important enzymes which are solvent stable and find applications in non-aqueous low water catalysis like peptide synthesis, transesterification, synthesis of organic compounds, etc. Piezophiles, radiophiles, metallophiles are also sources of enzymes which are stable under high pressure, radiation, high metal concentrations respectively, but given the difficulty associated with their cultivation, application of these enzymes are currently being investigated upon.
Thank you.
Thank you.